This lesson's about nucleosomes and epigenetic modifications. So your cells have a giant problem with how much DNA they have, because each cell has about 3 meters of DNA in it. And when you multiply that by the trillions of cells that you have in your body, you have enough DNA in your body that if it were all stretched out in a line, it would go from the sun to Pluto and back. That's how much DNA you have in your body. So there's an enormous packing problem of fitting all of that into a tighter space. And that's solved by packing the DNA onto chromosomes. And nucleosomes are the basic unit of structure in the chromosomes of eukaryotes. They are made up of a core of 8 globular proteins called histone proteins, with DNA wrapped around it. So they look like this. You've got 8 of these proteins, and then around that you have DNA that wraps twice in a coil and then comes out the side, and then that's repeated. And these 8 histone proteins are made up of pairs of identical proteins. So there's 4 different types of histone proteins in the nucleosome, twice each. Histones have positively charged amino acids in them, and as a result, they bind to the DNA quite tightly. Remember that DNA has a sugar phosphate backbone that has a negative charge, so that holds them together, the attraction of the positive amino acids in the histones and the negative charge in the sugar phosphate backbone. The tails of the histones extend outwards and can have functional groups added onto them, and we'll talk about that a little bit later. And by tails, I mean that these are proteins made up of polypeptides, and one end of the polypeptide is extending outwards. That's the histone tail. There's also a 5th type of histone protein called histone H1 that links the nucleosomes together into the next level of packing. So let's take a look here over on the right and see how this works. This is a diagram from Albert's Essential Cell Biology. You've got a DNA double helix here, and this is the nucleosome that I've drawn over here, shown as this kind of cake shape with the DNA around it twice. And that's repeated. And then these are bundled together, and the H1 protein here is what links them in this next layer of organization, this next level, and then these loop back and forth, another level of packing, and ultimately we end up with what we think of here as a mitotic chromosome, like a metaphase chromosome. And as you can see here, that's about a 10,000-fold shorter length than we started off with because of this packing. And that's how we get from DNA that could go from the sun to Pluto and back to all fitting in our body. So nucleosomes solve this packing problem. They're also involved in the regulation of gene expression, meaning determining when genes can be read out into messenger RNA and protein. Because enzymes for transcription can't access tightly packed DNA. So when the DNA is all bundled up as shown up there, you can't actually get the RNA polymerase in order to transcribe it, and so you can't make protein. But modifying the histones or modifying the DNA can either loosen or tighten the DNA structure to let those enzymes in more easily or to keep them out. And that'll therefore affect whether transcription can proceed. So for example, you can add methyl groups to the DNA. That's called DNA methylation. A methyl group is a CH3 group. And that will actually tend to silence the gene, shut it off, and prevent transcription, especially if they're added to the promoter. You can also add acetyl groups to histones. That's called histone acetylation. And an acetyl group is a C double bonded to an O double bonded to a CH3. And this actually neutralizes the positive charge. And by neutralizing the positive charge on the histones, then the histones will not attract the negative DNA as much. And so you get more loosening of the DNA structure and more access for the transcriptional enzymes. So this tends to activate those genes. And in fact, these kinds of changes, like DNA methylation or histone acetylation, can actually be transmitted to daughter cells during mitosis. And so a cell can pass this pattern of switched on and switched off genes to its daughter cells, and the pattern of gene expression is maintained. And this is called epigenetic inheritance. Epi means on top of. And this is happening on top of the genes, on top of the genome. The DNA nucleotide sequence isn't changing, but these differences in gene expression are being inherited. They're being passed from one cell to its daughter cells because of this passing along of the modifications to the DNA and the histones. And this is actually how cells maintain differentiation in a tissue. If you think about a liver cell dividing and then making daughter liver cells, remember that all the cells have the same DNA, and so it's not that a daughter liver cell knows to be a liver cell because it's got different DNA than an eye cell or a skin cell. Rather, it's that this pattern of differentiation is passed along by certain genes being switched on and switched off through this kind of epigenetic inheritance. So the daughter liver cells are getting the same pattern of epigenetic modifications that the parental liver cell had, and therefore they're becoming more liver cells. This is only one of the ways that this happens, but it's an important way. So that's an introduction to nucleosomes and epigenetic modifications.